to this workshop. So it's the third installation. I said we had like 2 workshops already. We organized this every 2 years. First one was face-to-face, second one online, and now face-to-face back again. So it is a workshop we put under the theme of state of the art. Basically, the general idea is not looking too much in the future, but rather looking in the present. So what do we have as tools? Are these tools like valid? Can we use them? You know, do we have enough experimental control to be able to make valid inferences on like causality of brain functions? So for this, we have organized these 2 days, and they are exciting 2 days ahead of us, I think. So we have the first day. Sorry, that's actually not the one I wanted to show, but it's okay. The first day is more on state of the art. So we have 4 speakers with like insight or overview presentations. We have a lot about electrical brain activity, I think. I'm not sure how much on electrical brain stimulation, not sure how much on magnetic brain stimulation, but also for the first time, we have a talk on transcranial ultrasound stimulation here at the workshop. That's really exciting. And then the second day, we are more specialized. We're having 4 talks on TMS and EEG. And at the end of each day, we have talks on multi-center initiatives. The first day will be on the Tox Challenge, 1 hour, an update. And the second day, we have presentations on other multi-center initiatives. And we think that these multi-center initiatives are really important, obviously, to have our tools ready in order also to make these causal inferences. We have organized the workshop so there's a lot of space to interact, space to refresh, space to profit from Italian pastry and coffee. So the talks are 1 hour, and breaks in between is 1 hour and a half, 1 hour to 1 hour and a half. And that also leaves us a lot of time to obviously look at the posters. Then as well, a small word to Rovereto. Those who do not know Rovereto, is a small little quaint town. It's like small enough so it's quite compact so we don't get lost, but it's big enough there's a lot of good restaurants and bars, so we can obviously also continue discussions then in the evening and meeting in the bars. I think that's what I wanted to say. A few operational points. It's about those who have not registered, who do not have a badge. So we are limited in seats here with 150. So if you see that there's no seat left, please let these seats for those who have a badge. There is an overflow room. It's the room 11, just next door. So you can go there and watch the presentations as well. Then the second point is about the posters. So we have like a poster session A and B. Poster session A is today and B tomorrow, but you can already put up your posters and then just be at your poster at the respective day. And as I already said, we really would value, in particular in this workshop, like this multi-center and collaborative work. And so we have these sessions at the end of each day where projects are presented that are collaborative, the Tox Challenge in this afternoon, and then 2 other projects in the afternoon session, the late afternoon session tomorrow. So there's a T4TE project, a NIPS-BITS proposal, and the proposal on the shared TMS questionnaires. If you have other ideas or if you have ongoing collaborative projects and you want to let others know, please let us know. We have still space in this hour. So 7 minutes, we can allocate 7 minutes, I think. That's it. Did I forget something? Okay, good. Okay, super. That's as a general introduction. So we're starting with the program then, and we have our first speaker, Gesa Hartwigsen. So Gesa is a cognitive neuroscience and neuropsychologist interested in plasticity neural networks for cognition, particular with an emphasis on language. She's using brain stimulation in combination with many neuroimaging techniques. She has trained as a PhD and postdoc in Leipzig and Kiel, has then been appointed a junior professor in 2013 as a professor of biological psychology at Kiel University, has then been appointed group leader at the Max Planck Institute for Human Cognition and Brain Science in Leipzig in 2015, and is now very recently appointed professor of cognitive and biological psychology at Leipzig University. And you will talk about, Gesa will talk about inferring causality from non-invasive brain stimulation. So, thanks for coming and thanks for...
Yeah, thanks for the invitation to this great place and for the kind introduction. So it's a great pleasure to kick this workshop off and share some of our attempts to infer causality from non-invasive brain stimulation experiments in cognitive neuroscience. And can you hear me? Good. And I will here mainly focus on TMS experiments in the language network. So when we're talking, about causality with non-invasive brain stimulation, then a general ratio or what we want to get out from a cognitive neuroscience experiment with brain stimulation is often the ratio that causality is inferred if inhibitory neurostimulation perturbs a certain brain area that we're targeting and has a measurable impact at the behavioral level. So as illustrated here for the example of TMS, we target a specific brain area, and ideally this will result in a decline in my task performance, in my task of interest, but hopefully not in a control task. So this is usually taken as evidence that this area is causally relevant for my talk, and this has some specificity. It may not be relevant for a control task, and ideally this would also have some anatomical specificity. So causality here means I inhibit a certain area, I get an induced disruption of performance. This means this area is causally relevant. So far so good. This can be termed as the conventional behavioral approach, and it will allow to probe the functional relevance of task-related activities. So we switch here. Hello. Much better. So we can, for example, first localize our activity with fMRI, then we target selected brain regions, and if we get a measurable impact on our behavior, we think that this area is relevant. One limitation of this approach is that we can only target selected areas. So we might use multifocal stimulation to target more than one area at the same time and infer causality about their interaction, but we cannot draw any conclusions about the underlying changes at the neural network level. And my group is particularly interested in the stimulation-induced plasticity at the network level. So how brain stimulation modulates not only the behavioral output, but also the underlying neural network activity and network interactions. And if you're interested in such question, then a good approach is to combine neurostimulation with functional imaging, as illustrated here. And I think across the course of the workshop, we will also hear more about the combinations of neurostimulation and EEG. I will focus here on fMRI and on TMS because this is the main methods that we are currently using in my group. So the idea is that we can use neuroimaging as a readout to map stimulation effects of plasticity-inducing brain stimulation protocols, for example, rTMS. And one idea is that we can target brain areas with inhibitory rTMS or also facilitatory one, and then map the consequences of our perturbation or facilitation with fMRI afterwards, which gives insight into so-called short-term reorganization or plasticity that is the response of the brain to a modulation induced by plasticity-inducing neurostimulation. We can also combine this in a simultaneous fashion, for example, with TMS directly during fMRI, which will give us insight into the immediate responses at the network level. And I think the beauty of this approach, and why we are using is that we can probe the impact of neurostimulation at the neural network level. So we can make inferences about changes, stimulation-induced changes in task-related activity, in task-related interactions, and relate this to the induced changes at the behavioral level, and thereby hopefully get more insight into what neurostimulation actually does. And as we will see, the effects are not as focal as one would always expect. Especially for plasticity-inducing protocols, we see strong remote effects, strong network effects beyond the area that we are targeting. So my group is mainly interested in language as a core function of human interaction and communication, and we're particularly interested in the flexible neural network recruitment during different kinds of language operations. This is data from a recent meta-analysis that we're currently publishing that was led by Sabrina Turka from my group, who's also here, and which illustrates from a number of hundreds of neuroimaging studies on fMRI-related language neuroimaging studies, that language is organized in distributed regions across our brain. So we may have left hemispheric dominance, but also strong contributions of right hemispheric regions of the cerebellum and subcortical areas. So we have a network here. And we find that specialized networks can be dissociated, which contribute to different linguistic operations, such as the semantics or the processing of the meaning of our words, and its phonology or the processing of sound of words. And now the key thing is that successful communication requires flexible interactions between these specialized subnetworks. And what we are interested in is the question how the language network may adapt to perturbations. Perturbations can occur in everyday life under challenging listening conditions. For example, when you speak in noisy environments or try to extract what someone is saying in a noisy environment, as the microphone that I had before. Perturbations can be induced in a controlled way by using inhibitory neurostimulation. And perturbations also occur in a number of disorders. For example, if you have network disorders, as we will see later, and also after brain lesions, such as tumors or stroke. So, big question, how does the language network adapt after such perturbations, and how does this help to maintain or recover function? So what do we know? What have we learned from neurostimulation studies in the language domain that address this question of adaptation in response to inhibitory neurostimulation? This is an overview from a recent review where we summarized a number of studies that found that perturbing one key language area or auditory cognition area, mainly in the left hemisphere, with inhibitory neurostimulation results in a stronger recruitment of the contralateral homologous area. So we see that after perturbation of a key language area, we see a stronger upregulation, so stronger task-related contribution of the homologous area, mainly in the right hemisphere, which is often interpreted in terms of compensatory activity increases, which help to maintain task function at a high level. So the idea is that perturbing the dominant hemisphere may result in a stronger contribution of the contralateral homologous areas, mainly in the right hemisphere. Now, based on such neurostimulation studies and many other neurostimulation studies, I have recently suggested a novel model for flexible redistribution in the language network. And the model is relatively simple and has two key assumptions, namely that our language network can adapt either by flexible adaptation within a specialized network or by adaptation across different networks. So how should such flexible adaptation within a specialized language network look like? Let's assume we have a simple network illustrated here, and we perturb one of the key regions with inhibitory TMS. So we decrease the relative contribution of this area to our language task. This will also decrease the task-related activity in this area, so this area is contributing less to the task. Now, how should the network react? What we sometimes observe in neuroimaging studies that map the consequences is an upregulation of the remaining network regions. So this would indicate a certain robustness of the network. The rest of the network simply works harder to perform the job. Alternatively, or additionally, we sometimes also observed a shift of the functional weight towards other areas of the network that may not have strongly contributed before our perturbation. So this could be regions that have been at threshold before and now work harder to be able to maintain function. And finally, as illustrated before, we also see upregulation of homologous areas in case of language in the right hemisphere after perturbation of left hemispheric key regions. The contribution of such homologous areas, especially after brain lesions such as stroke, is still widely debated, but we have some evidence that in some cases there may be a beneficial contribution. And I think the main take-home message is here that such flexible adaptation in the contribution of these different areas within a network is highly adapted and may compensate for the induced dysfunction of neurostimulation or also potentially after brain lesions. Now, in case of large-scale disruption, let's assume we target a key node that is densely connected with the rest of the network and we see a strong inhibition across the network. Then, of course, these regions may not be able to compensate for the network disruption. And in such cases, what we sometimes observe in neurostimulation studies is the recruitment of alternative networks. Alternative networks could be neighboring networks for other specialized functions. For example, in the language network for a different linguistic operation, when we target the semantic network, we may see stronger contribution of the phonological network. And what is currently of greatest interest to me is the recruitment of domain general networks. So domain general functions such as attention, working memory, or cognitive control are known to contribute to all higher level cognitive processes, including language. And the idea is that if I have large-scale perturbation of a specialized network, then the recruitment of a domain general network may to some degree compensate for the impairment and help to maintain or, after brain lesions, regain function. And I'm not going to suggest that a domain general network can replace the function of a specialized network. I'm fully aware that this is not the case. But the main idea is that a stronger contribution of such general resources is adaptive and a helpful mechanism of compensation. Now, two big questions remain open. The first is the behavioral relevance of such shifts in activity patterns. It's nice that we see changes in task-related activity after perturbation, but a really open question is, are they meaningful and are they related to behavioral relevance? So do they really causally contribute to my behavior of interest? This question, I have to spoil this a bit, has to remain open, but maybe there are some ideas how to address it. And a second question is whether these adaptation mechanisms can be transferred or generalized to other cognitive functions. So is this flexible adaptation in cognition a general principle that generalizes beyond the language domain? So let's come to the first question first. What about the behavioral relevance of this shift in adaptation, in the recruitment of different networks? I think currently from most experiments that we see, this has to remain unanswered because it would require designing a new way of experiment or an ideal experiment to probe compensatory reorganization. And this would look as follows. First, we would need an fMRI experiment to localize the activity of interest, for example, a language localizer with fMRI. Then we would target a key area, for example here inferior frontal gyrus and perturb this function, and afterwards map the consequences with fMRI. Ideally, we might see inhibition at the targeted site and maybe some compensatory upregulation of other areas. This is where currently most studies stop. However, to really make the claim that this upregulation is causally relevant and really contributing in a compensatory fashion to my behavior, we would now additionally need to perturb this reorganized region and then map the consequences again of this double perturbation of my area of interest and the upregulated area. And with this, I could infer changes in task-related activity and behavior after perturbing my area of interest and the observed compensatory upregulation. So the ratio would be, if the observed upregulation or reorganization that we see here is compensatory, additional perturbation of that region should further impair performance and task-related activity or network interactions. I could stop here and say that this has, to my knowledge, rarely been done in the study of cognition. So the majority of studies focus on localizing activity, perturbing single areas, and then map the consequences. But I think we can still learn a lot from this, what might be regarded as state of the art here. And I will come back to this idea of going further in causal inference later. So what about the second idea? Can we generalize these mechanisms of flexible network adaptation beyond the language domain? So what I want to show you here is some examples on flexible neural network recruitment beyond language for other cognitive domains, and especially focus on the role of domain general networks in cognition. So this is work led by Ole Numsen from my group, who's interested in domain general network interactions across different cognitive domains. And he focused on the contribution of the inferior parietal lobe, which is a heteromodal association area, which is associated with a number of different key domains of human cognition, namely attention, language, and social cognition. So Ole designed an fMRI experiment where he had 3 tasks of these 3 domains in the same subjects, and he could show that this larger IPL region can be parcellated in specialized subregions that contribute differentially to these tasks of interest. And what you can see here is that in both hemispheres we can distinguish an anterior and a posterior region. And now the interesting thing is that the predictive relevance of the task-related activity in these specialized subregions differs across domains. So we found the strongest predictive relevance of task-related activity for the attention task in the right anterior IPL, whereas our semantic language task had the strongest predictive relevance in the left anterior IPL, and our social cognition task had the strongest association with both posterior subregions. And why is this interesting? Because I think that this shows that even if I have heteromodal regions, by designing good fMRI experiments, I can distinguish task or domain-specific contributions and specializations of different sub-areas. And finally, we could also show that these areas also differ in their task-related connectivity profiles with the rest of the brain. So it's not just this area itself, but also its interaction profile with the rest of the brain that is highly specific for each task. So what's illustrated here is the connectivity profile for social cognition, where we found both posterior subregions to be densely interacting with major brain networks that have also been described as REST, so with other areas of the default mode network, dorsal attention network, control network, and so on. So the take-home message is that there is a specific contribution of different IPL subregions to different cognitive domains. And the obvious question is, what happens if I perturb these areas or some of these areas? Will I also get task-specific changes in the response profiles to neurostimulation? And may there be some overlap, some general response mechanisms and some task-specific mechanisms? And this is work led by Katie Williams from my group. She's also here, and if you're interested in the details from this project, please visit her poster. It's number 43, and it's about the perturbation of the IPL and how this induces task-specific changes in network interactions during different cognitive tasks: attention, social cognition, and language. And to give you a brief overview of what we've done here, so we targeted the posterior subregions in our IPL with inhibitory TMS, with CTBS here, and then mapped the induced changes in task interactions at the network level. So this is network interactions derived from an independent component analysis across the 3 tasks. And we then used a correlational PPI approach to see how neurostimulation affects the large-scale network interactions. And you can see that there are different changes in the interaction profiles for the 3 different domains. And I think the most simple take-home message here is that the more complex cognitive processes, also in terms of the behavioral complexity, which you can measure with your response times, that is the social cognition domain, showed the highest responsiveness to neurostimulation. So we have only a few interactions that are influenced by neurostimulation for the simpler tasks. So for our attention task, we only see neurostimulation effects when targeting the right hemisphere, which is in line with what you would expect. For semantics, we already have a bilateral pattern, and for social cognition, we also have a bilateral pattern of the more complex changes in task-related interactions. What is interesting here to note is that these changes seem to depend also on the domain in whether they induce inhibition between the network interactions or facilitation. And one side note, these interactions mainly always engage the domain general default mode network. And I think if you want to know more about the details, please talk to Katie at her poster. But the take-home message is that we can also induce task-specific changes in large-scale network interaction here with inhibitory neurostimulation. And I think that may give some insight beyond we see a slight change in task accuracy or in task-related response speed. Now, a second topic that we are currently very interested in terms of flexible neural network recruitment is how this may change as we age. And in particular, we're interested in the role of domain general network interactions in the aging brain. Why is this of relevance? We know that as we age, there's a strong change in the recruitment of domain general networks and resources for cognitive control. And it's hotly debated whether the changes that we observe, some of them already addressed, are beneficial and help to compensate for cognitive decline, or may rather reflect the decline itself and reflect dedifferentiation of networks. So this is a work led by Sandra Martin from my group, who's interested in neural reorganization in the aging language network. So she used a verbal fluency task, which is a highly ecologically relevant function because we often need to search for words that we need to retrieve. And Sandra found increased interactions in domain general networks to be associated with better behavior in older adults. So this is first data from a neuroimaging experiment where she compared older adults with young adults during verbal fluency. And what you can see here is that older adults generally show increased task-related activity in domain general areas, especially in these midline structures that are part of the multiple demand network for cognitive control, so particularly pre-SMA and underlying cingulate cortex. Older adults also show increased task-related interactions between domain general networks. Here you can see a seed region from a psychophysiological interaction. In the right insula, a cognitive control region, which showed increased interactions with areas from the default mode network. And finally, most interestingly, the increased interaction inside or within this domain general multiple demand network seem to support behavior in older adults. So only in older adults we found a correlation of the strength in the multiple demand network connectivity with better executive functions. So it seems that this change in the connectivity profile in the aging brain is associated with better overall cognitive performance selectively in the aging brain. But of course, now the question is that also relevant for our language behavior, not only for general tasks? And indeed, when we reanalyzed this data with a network approach, then we found that we can also see increased facilitatory network interaction to be associated with better verbal fluency. So what you can see here is the age differences in the network interactions. And again, we took here the task data from verbal fluency during fMRI and performed an independent component analysis and then a correlational PPI approach to investigate large-scale network interaction. And the most striking pattern here is that increased facilitatory network interactions between task-positive and task-negative networks is only observed in the aging brain. So in aging brains, we see that networks which are normally anti-correlated now interact more strongly, whereas these networks, for example, default mode network and attention network, are anti-correlated in the young brains. And this seems to be behaviorally relevant. So the facilitatory network interactions are associated with better verbal fluency selectively in the aging brain, whereas the opposite pattern is true in the young brains. So here it seems that anti-correlation is reflecting the normal, the better behavior. So it seems that this shift in the activity pattern, stronger interactions between networks that are usually anti-correlated during a language task are associated with better behavior as we age. And this is also supported by data from a graph theory analysis where we could show that older adults also show less segregation between networks. So segregation is reflecting the specialization of a specific network. And older adults relative to young adults show reduced segregation of these networks. And it seems that this a strong degree of segregation is also associated with better language accuracy and better executive functions outside the MR scanner selectively in the young brain. And I think together this data implicates that there is a shift in the strategy in the aging brain, namely more interactions between networks which are usually anti-correlated, and this shift may reflect an attempt of the brain to compensate for the overall cognitive decline and adapt to changing conditions in our brain. And now, finally, we thought if this contribution of domain general networks, especially multiple demand network, is beneficial in the aging brain, then targeting a keynote from this area with facilitatory brain stimulation may help to improve behavior and may also shape the network interactions towards a pattern that may look a bit more youth-like. So maybe in this case we could use facilitatory brain stimulation to improve function. So we targeted the pre-SMA, this was this area that showed strong upregulation in the aging network with ITBS here, and then afterwards performed a semantic judgment task. So we had to find a task that had required some degree of semantic control during fMRI. And I have to say that the results were rather inconclusive. So at the behavioral level, we did not find anything. The results were super heterogeneous. Some older subjects got better, some got worse, the majority did not show strong effects at all. And also the results on task-related activity are quite puzzling. So we found that facilitatory TMS increases task-related activity, but not in the stimulated area or in semantic language area, but only in remote regions of visual and ventral attention networks. So here you can see the stimulation sites, and for semantic processing, so for our task of interest, we only found remote activity increases in more general control resource and attention networks. We also found changes in the task-related interaction patterns, so in connectivity. This is data from PPI analysis, where we used the areas at seed region where we found these increases in activity, and then looked at induced changes in the interactions with the rest of the brain. And this is overlaid on the parcellation of U, where you can see the different networks. And the main pattern here is that ITBS of the pre-SMA decreases the task-related connectivity mainly in domain general networks. So we have an increase in activity in remote regions, but a decrease in connectivity. And to make this even more complex, we found, when we looked for correlations with behavior, we found that increased coupling between the dorsal attention network, so between a seed region where we could see also changes in activity, and the pre-SMA, so our target region, was related to more efficient semantic performance. So it seems that a specific change in the coupling between our target region and an area for attention is associated with better performance during the semantic task. And I think these quite puzzling results show that the results that we get are not always as straightforward and not always in line with our hypothesis, but network analysis may capture the more subtle neurostimulation changes even in the absence of behavioral effects. And the effects that we get on task-related activity don't equal what we see in task-related interaction. So we may see an overall increase in task-related activity, but a decrease in task-related connectivity. And I think it's important that we relate these stimulation-induced changes to behavioral changes, because otherwise it's really hard and complicated to interpret them. But I wanted to use this example here to illustrate the heterogeneity of results and the difference between induced changes at the stimulated area where we didn't find anything and in strong remote effects, plus activity changes do not equal connectivity changes. Now, finally, I want to briefly discuss neural network recruitment in network disorders and the role of domain general networks here. There's already some literature on the recruitment and potential compensatory recruitment of domain general networks in language recovery after stroke. But here I decided to choose an example of a network disorder, a learning disorder that is related to reading impairments, namely dyslexia. So what do we know about dyslexia? This is again a study by Sabrina Turka from our group, who was interested in network interactions during reading in adults with dyslexia. So dyslexia is a relatively frequent reading impairment that is usually coming up during development, but can persist into adulthood and results in reading and writing impairments. And Sabrina did a fMRI study first where adults with dyslexia, had to read words and pseudo-words, so non-existing words, with increasing complexity and were compared against healthy controlled subjects. And what you can see here is that these dyslexic subjects show an underactivation of task-related activity during reading, which is extending beyond classical reading areas in the left hemisphere, like the left supramarginal gyrus and ventral occipitotemporal cortex, and is also manifested in underactivations in the right homologous area and the cerebellum, and also a number of domain general areas. So this hypoactivation or underactivation of the reading network has frequently been described in the literature. We show that this extends beyond the classical reading network. When we look at changes in task-related connectivity between these areas that are underactivated, then we see that it's also nicely reflected in a pattern of hypoconnectivity. So even if this looks very complex and detailed, the message is relatively simple, namely that adults with dyslexia also show reduced hypoconnectivity between reading areas, extended reading network areas, and the rest of the brain. And the most striking pattern was found for the right cerebellum. So the right cerebellum shows a strong decrease in its interactions during reading with the rest of the brain. So we think that this hypoconnectivity may be a way to understand this reading impairments in adults with dyslexia. So to summarize this, we find that dyslexia is characterized by underactivation and hypoconnectivity within and outside the classical reading network. Using effective connectivity analysis, we could also show that these changes or aberrant connectivity in the reading network and beyond is behaviorally relevant. So we think that dyslexia can be characterized as a network disorder, which is characterized by strong aberrant functional connectivity profiles. And we then designed a neurostimulation study and addressing the potential of facilitatory RTMS to change these underactivation at the network level and potentially also the network interactions and improve behavior. And I will not present the details here, so if you're interested in the potential of facilitatory brain stimulation in dyslexia, please visit Sabrina's poster. So I think for the rest of my talk, I want to focus on the way forward. So I think we have some ideas about flexible adaptation in response to neurostimulation, both in the healthy young brain as well as in the aging brain, and potentially also in network disorders, but the results are often not as straightforward as one would wish. So we often see quite heterogeneous results and puzzling patterns, for example, no effects at the stimulation site, only strong remote effects. And one question is, how can we improve this? How can we improve the causal inference that we can draw from non-invasive brain stimulation and also the stimulation efficiency? And I think one way is, and now I come back to my initial thought about designing the ideal experiment, is that we need to probe the causal relevance of the flexible adaptation at the network level. And this is a project that I will start next year, which is called FlexBrain. And that directly addresses this question by using a network stimulation approach. So the overall idea is that we need to map flexible adaptation across networks by targeting more than one node at the same time. I think especially if you look at complex functions such as language, then it's maybe too simple to just target one area and look at induced changes. What we want to do is target simultaneously several areas of the specialized semantic language network, then use fMRI to map the induced changes, and hopefully see that if we target more than one node, we see a strong inhibition of the semantic network and potential compensatory upregulation of domain-general cognitive control networks. And then the interesting thing comes into play, namely that we probe the causal relevance of this reorganized pattern by combining stimulation of the specialized network, semantic network, with online perturbation of the reorganized pattern directly during fMRI, so to use simultaneous TMS in the scanner to probe the causal relevance of this observed upregulation. And this will require a complex combined perturbation of at least 3 regions, parts of the semantic network, and 1 or 2 regions of the upregulated domain-general network. And I think this may ultimately give some or might provide some evidence if the observed upregulation is indeed causally relevant for language behavior and also for the network interactions. So another way to move forward is to improve the stimulation efficiency itself. And I think one good way that is increasingly being used is to use electrical field modeling. And this is work that we're currently doing with the group of Thomas Knösche at the MPI, where we are very interested in getting a better understanding where we stimulate, where we should stimulate, and how we should dose our stimulation intensities. And I have two examples here, one of a post-hoc modulation of the E-fields and one of an a priori simulation. So this is work led by Philipp Kuhnke, who's a postdoc in our group and who's also here and will also present something, and who used E-field modeling to identify the areas in the brain that were effectively stimulated. So Philipp had a conceptual semantic processing experiment, and he found that if we target the left inferior parietal lobe, we get a decrease in the task-related accuracy. And then afterwards, we used E-field modeling to show where effective stimulation took place. And you can see this outlined area was obtained from an fMRI experiment, and this is the area of the IPL. And Philipp could show that the individual E-field strength predict the stimulation-induced disruption. That is, stronger stimulation of the IPL region is associated with worse performance during conceptual processing. And this association, this correlation, was only found for the region of interest in the IPL, not for neighboring regions that also received relatively strong stimulation as shown by the E-field, here for example in the somatosensory cortex. I think this is a first way of improving our knowledge about what we're doing in the brain by showing where we effectively targeted our brain area or our network. Of course, ideally, we would do this a priori before we start the experiment, because otherwise it may be too late. And now this is what we're currently doing, so to use a priori field modeling to optimize targeting and dosing. And here again, if you're interested in the detail, please visit Phil's poster. I will only briefly mention what this may be about, namely that the standard ratio in studies of cognition and neurostimulation studies is mainly to use a fixed intensity, for example, 100% of the individual resting motor threshold. Now, we thought that the optimized intensity should be corrected to induce the same electrical field as we would induce in the M1 motor hotspot, where we get our motor threshold from. I know that there are previous attempts to correct for the scalp to cortex distance, but we thought that we should correct for the induced electrical field. And this is how this looks like. So here you can see E-field simulations where the magnitude of the electrical field is regressed on the MEP amplitude from my motor evoked potential. And this can be used to optimize the position of the coil to get the strongest E-field in the area of interest in the motor cortex. And this may also be transferred to areas outside the motor cortex, such as the IPL. And to foreshadow these results, what we find here is that the corrected or optimized intensity may strongly differ from this ratio to just use 100% of individual resting motor threshold. So what this should illustrate is that we should individually optimize our dosing and also our targeting based on the E-field distribution that we can simulate before our experiment. And why might this be of interest? Because the cortical folding patterns have an individual and a strong impact on the functional discriminability of our TMS. This is again work by Ole Numsen, which illustrates that often we want to distinguish the contribution of specific subregions in a larger area to a given language task, here illustrated for Broca's region. And you can see that if we target this area, we get the strongest E-field under our target site, which is good, only a slight E-field here in the neighboring region. Whereas if we target this area here, we also get a strong E-field in a neighboring region. And then we may see a behavioral effect and think, oh fine, we have a disruption, but this may also come from a neighboring region. And finally, if we have an area that is located quite deeply, so here, we might not get the strongest stimulation effect in terms of the E-field in the target area, but only a neighboring region. And this is just impacted by the individual anatomy and the individual folding. So take-home message, electrical field modeling can help to identify the areas that are effectively stimulated. And I think this is particularly interested if you're interested in the dissociation of neighboring regions and want to perform functional anatomical double dissociations of neighboring subregions. And also it is interesting to know the relative stimulation intensity that is received in each of these areas. So to summarize, what food for thought may I give you on flexible neural network recruitment during human cognition? I think everyone would agree that neural networks must remain flexible to adapt to challenges. We see that network recruitment patterns depend on specific domains, and perturbation induces domain-specific changes in large-scale network interactions for different cognitive functions. We also see that neural network interactions change with age, and the big challenge is to dissociate patterns of compensation and general de-differentiation to know what is beneficial and what may be maladaptive. We see that increased demands require more domain general contributions and interactions, and maybe also less segregation between networks. We also see that aberrant neural network interactions for reading impairments, so maybe reading impairments can be characterized as network interactions based on aberrant network connectivity. And in particular, we think dyslexia is characterized as network disorders, and this should also be included in the treatment. What should be done now is to explore the potential of facilitatory neurostimulation in these conditions in aging brains, lesion brains, and of course for network disorders. And I think a way forward is to consider a more network perspective to target more than one area and probe the causal relevance of the interactions across different networks. And finally, electrical field modeling may be used to optimize our targeting and dosing and be more precise and get a bit more homogeneous results in the future. Yeah, I think that's it. So thanks to my group for the wonderful work. And yeah, I'm looking forward to discussing all this now.
Thanks a lot, Gesa, for that impressive talk. So we have plenty of time for questions. Maybe I start with a question. So towards the very end, you mentioned maladaptive, and you didn't really, I think, introduce it before. Obviously, this compensation might all be maladaptive, and so sort of like the second intervention might, and if you want to improve function, might actually be, or it might be of interest to inhibit again this, so in order to improve function.
Oh, yeah, I use, no, I take that. Ah, okay, so for the audience. So this is speech and noise now. Now it's gone. Yeah, so the question was about maladaptive plasticity. What is this, and can we also potentially use non-invasive brain stimulation to counteract or prevent or treat maladaptive plasticity? I think this is a very interesting question, especially in terms of stroke-induced impairments, which I didn't discuss here. So one of the big questions in the stroke recovery literature is if we see that after stroke in the left hemisphere, we see language impairments, and then we see an upregulation, stronger contribution of these homologous areas in the right hemisphere. Is this reflecting compensation, or is this reflecting maladaptive, that is misleaded plasticity that is hindering the recovery of function? And there's evidence for both. So some studies find that it seems to be adaptive, others think it's maladaptive. I think the answer is it depends on the time course of recovery. Here in the stroke example, early upregulation may be adaptive, later upregulation may rather reflect maladaptive plasticity. And there are some attempts that if this is maladaptive, then I should inhibit these areas. So the idea is if that the upregulation of the right hemisphere persists into, let's say, the chronic phase after stroke, I can use inhibitory neurostimulation for treatment and reduce this maladaptive plasticity and reshift the activity back to the left hemisphere. And I think this is a quite interesting example or an interesting idea that may also translate beyond this example of stroke and plasticity. and may also explain some of the patterns in the aging brain. So some studies also find that not all these patterns or these shifts in the patterns that we see in the aging brain, also our own studies were not as easy as I showed here, some of these changes in the patterns and some of the stronger interactions between domain general networks are rather associated with worse performance, less efficient processing, and more errors. And here we might also think about using inhibitory neurostimulation to selectively or to inhibit these patterns of activity and reshift the interaction patterns to a more youth-like pattern. So I think what I presented here is, of course, super simplified, and it's only part of the story. And in the end, I think when we think about interventions, making training or cognitive decline interventions in aging and maybe also in Alzheimer's disease more effective, and also bringing this to brain disorders such as stroke, then what we may need is a mix of inhibitory and facilitatory protocols that should be applied in specific phases of the disease. So in stroke recovery, we may need, at least for language recovery, different protocols in the very acute and early phases than in the later phases. Yeah. So sorry for doing so much blah, blah, blah on one of my favorite questions.
Thank you very much for the beautiful talk. And I would have millions of questions, so which is good. But typically in these inhibitory protocols, which is either 1 Hertz offline TMS or theta burst, what is also interesting to look at is the long-term effects, because they can have important implications for treatments and interventions, as you mentioned. Yeah. So have you looked at any long-term effects, but not just in the changes on network, but also changes in behavior? Yeah. And my other question is, in the last part of your talk, you talk about e-modeling, the modeling of E-fields. Does this very nice and precise analysis of where to go and target with the stimulation correspond to a more stable behavior across subjects? Have you tested that? Thank you.
Yeah. So I try to remember both questions. The first question was about the more long-term effects, and I think here I have to give the disclaimer that what we are doing is only single intervention, so we apply a protocol just once. But I know from the aphasia and training literature that a number of studies also used RTMS protocols more than 1 times and found some effects that persisted beyond the end of stimulation. So there's a number of studies from the Mainzer lab where they showed that even up to 3 or 6 months after the end of the intervention, some of the behavioral changes were still persisting, also in patients with post-stroke language impairments, and some of the changes even needed some time to build up. So in one of their studies, they did not find an immediate effect, but only some months after the intervention. But when I look at the results, I think this is also quite heterogeneous. So some studies find no effects, others find a lot of variabilities. Some patients seem to benefit more on the long run. And I think this is also, may also be true for the aging brain, because it depends on the individual level, so on the level of task performance, why you get the simulation, that some may benefit and others don't. And as for the E-field effects, we're only beginning to explore this. So we don't have any long-term effects, so we haven't finished our study on cognition with this a priori E-field modeling to really show that this also reduces the variability at the behavioral level. I think so far it is safe to say from what we know from the motor system that we can capture some of the variability. But if this translates to making more efficient behavioral interventions with TMS or also TDCS, because the same mechanisms should apply here, that I cannot say yet, but I would guess so, because if we capture more variability, then we may also optimize the stimulation to the individual needs. Yeah. I think Till had a question.
Yeah, thanks, Gesa. It was a fascinating talk, all new data that I didn't know yet. I have a question on the remote network effects following offline TMS to a certain network node. Yeah. There are always 2 possibilities, right? Either you produce local plasticity, local changes in excitability that then has a network effect, compensatory or maladaptive or whatever.
Yeah.
Or during the offline protocol itself, we know that activity spreads,
Exactly.
right?
Yeah.
And it could induce plastic changes in the network, in the remote nodes already. So is there any chance, any experimental design you could think of to disentangle these effects?
Yeah, ideally, I think this is a super interesting question, and I think I should also mention that all these network effects that we get were only shown for plasticity-inducing protocols here. So first, I would hope that for online protocols, if you directly modulate task performance during behavior, the effects are not as strong, because otherwise, don't know, 80% of the conclusions that we draw from behavioral studies might be wrong or underspecified. And now coming to your question, I think an ideal experiment would use an offline protocol during fMRI, so to map maybe at rest during fMRI what we are inducing and how the networks effect change across time, and then continue maybe with the task. I haven't really thought about this before, but it's an excellent question. And then see how this translates to network effects on the behavioral level. And I'm not sure if this is possible. That would mean that maybe if we do 10 minutes of 1 Hertz stimulation in the scanner and then immediately afterwards perform a task, this could be possible. I don't know if the guys in the first row have already done something in this direction, but I think it would be an interesting question to know what are the plastic effects that we look at. Are these plastic after effects, that is the response of the brain, that's how I sell it, or may they to some degree already reflect the direct modulation, right? I have to say I don't know the answer yet, but maybe we get there.
Thank you.
May I add a simple technical question? You mentioned the inconclusive results of the unpublished Martin data and the SMA, pre-SMA stimulation. On the slide you showed an anterior-posterior coil orientation. I think you might have better results with a 90-degree turnaround.
Yeah. That we discussed a lot beforehand. So I think half of the studies that we found, and that also varied a lot across motor studies that targeted pre-SMA and SMA and cognitive studies, half of the study used this 90-degree orientation like this, if this is the nose, and the other half used this. And I don't remember exactly why we chose for this orientation, but I think it was in the end that the more cognitive studies, more of them follow this orientation. But I think this is an open question. Yeah, maybe we get better results if we do this.
I may generalize this question. I am maybe an overshooting addict of the cosine hypothesis. So the electric fields you showed in your last slides are purely physical fields. They have nothing to do, I bring it really provocative to a point, they have nothing to do with what is really entering the brain. So according to the cosine hypothesis, the tangential current flow of the TMS pulse, I will mention that in my talk, will simply pass along and does not enter the brain at the crown.
So I think I would turn this around. I would say the direct activity that we induce with the stimulation is completely underestimating the effective current that is reaching the brain. And I think that's why we need this E-field simulation. And I agree that we here in these simulations only used at the normalized component, and some of the other and at the magnitude, and some of the other components that may also strongly influence the effect that we get are neglected at the moment. But I think showing that these distribution of the electrical field that we have so far, and I would also say that this is just a rough estimation of what's going on, has a correlation with the induced stimulation effect that is also in line with what we observe from our fMRI results, already shows that this may go into a good direction. And the question is how we can improve these field simulation to make this more realistic or use more realistic models of how this, what this actually does in the brain. So looking forward to your talk, maybe you have the answer.
Hi, very nice talk. Thank you. You said that in the aging brain, networks that are normally anti-correlated can become correlated. And I think the example you gave was the default mode network and the attention network. Is there a cost to that happening?
What was the question? What the cost is?
Is there a cost? If a network that is normally anti-correlated, now two networks that are anti-correlated become correlated, is there a cost in behavioral performance to that? Yeah.
Yeah, I think the cost may be more efficient processing because, I mean, those, so in the study that we performed, the older subjects were not performing worse in our task, but they were slower. So the cost may be the efficient processing. So that it takes more time if the interaction architecture of our brain changes. Yeah. So I would think that there may be a cost in terms of efficient processing, but I mean, we don't know yet if the cost is outweighed by the benefits that we get, but because this is only, I mean, this is a correlation in one specific language task. So, and I think in one of the studies we found, yeah, we find this correlation for both language and executive function, but at the same time, we might also see that this does not generalize to other functions. Yeah, my guess would be that it may, that the cost effect may go towards the efficiency of the network interactions, and which may be reflected in older subjects needing more time, so in terms of response speed. But this is, it's a hypothesis, I don't know this yet, yeah.
May I add another technical
more e-fields.
No. We know from Antonio Oliveira's work that static magnet inhibits the motor cortex with quite a low field, around 0.1 Tesla. Yeah. Now, all these experiments in the scanner, the static field of the scanner may influence the brain's activity per se. Yeah. Do you have control experiments for that?
No, because then we would have to have an experiment without the static magnetic field of the scanner, right? But I think at least we can have that, I mean, this is controlled in a way that this is kept constant, right? So this should, we always have control tasks and control conditions, so the same should apply across all my tasks, unless we could assume that the static magnetic field of the scanner selectively affects my language task, but not my control task, which I think would be physiologically rather unlikely. Or is that not what you mean?
You could draft a quick experiment and then keep the subject in the scanner for 2 hours and then repeat that experiment.
I mean, yeah, but why? What do we want to show this?
I want to understand if 2 hours in a 3 Tesla scanner inhibits the brain. Mm-hmm.
Yeah, I don't see why this should have specific effect on a language network and not on other cognitive tasks. So I would think that this should subtract out, but I mean, feel free to do so. Also, I would think that since we normally do repeated sessions with different conditions, we may get rid or at least balance this effect out. Yeah, but in principle, interesting idea, I would think. Not sure if this will be the next experiment on my agenda, but maybe on yours.
Thank you very much. I'd like to go back to your experiments with pre-SMA and the effect you found in the attentional network. It perhaps could be necessary to target the attentional network afterwards to see, in terms of causality, is the title of your work. Why you haven't done that?
Because it's so much work. I mean, it sounds so simple, but yeah, I completely agree. So if you didn't get the question, the idea was for this inconclusive pre-SMA stimulation, if we see this restructuring or stronger interaction with this attention network, wouldn't it make sense to target this network now and see if this affects behavior? And I think this is a very good idea, and this should definitely be something that we should do. But it sounds so simple that we should just run another session, but if you think this through, then we would have the same experiment again, and add the additional condition that we then target this upregulated or reorganized network. This is exactly what we want to do. I'm not sure that we will use this particular task for it, but this is exactly what we're planning to do now, because so far I think all the conclusions that we, and I include myself, now draw about this, oh, this is compensation, it's related to the absence of any effect, right? So that's why compensation is so hard to really prove. We should show that if this is compensation and then we target this, then we should make behavior worse. And this is something I think that yet needs to be done. There's some work on this in the motor system, but to my knowledge, not what we would actually need in the study of cognition now. Yeah.
A very fast, quick question, technical question. Continuous theta burst is normally very variable, render very variable results. Have you found that in your data?
The variability of the rights?
With theta burst.
Yeah.
And how you deal with that?
Yeah, we find that always, and this is super frustrating. As I said, that for some of the subjects we see the effect in this direction, for some in the other direction, for some no effects. So sometimes we just don't have significant behavioral effects. I think the way to deal this is look at individual subjects and include that in the analysis also at the behavioral level. So for example, if you use mixed model approaches, then you can also account for the individual variability. But I think I've been recently very discouraged from using theta burst at all because the effects are so variable. So we're now moving back to the good old 1 Hertz protocol. But as a spoiler, in some of our most recent studies also that was very variable, and also for tasks much simpler than language, we did not find significant modulation and quite strong heterogeneity. So,
thank you.
Yeah, I don't have a good answer. If you know it, let me know then.
No, I don't.
Okay, we take 1 or 2 questions more, and then we go for coffee, I think. 3 questions.
No, you go, and then It's all right. You mentioned that you would not expect the same effect for online paradigm, and this is, of course, very important for all the studies that use TMS without neuroimaging. So I was wondering, what would you expect? Like, would you expect like nothing like this, or would like this is well unlikely, or do you expect to find like some of the effects that you found? Like, and the implications then for
I think that's a very good question. So the question was if we would expect the same effects or different effects for online protocols. So my hope would be, I mean, the ratio I think is different. If we use offline stimulation, we induce plasticity, the brain has time to adapt all that. And for online stimulation, we would hope that the effects would be more focal. But I think from the studies that we know on combined concurrent TMS and fMRI, we know that this is unfortunately not true. So already in the motor systems, we see a lot of spread of the activation. And maybe this is also maybe this is a good indication, because this is why we have some stimulation effects. If it would be super focal, maybe it wouldn't even be strong enough to induce anything at all. So I might expect, and I mean we will see that hopefully soon, that the effect get a bit more focal, and maybe we don't really see these plastic effects, these compensatory effects, but maybe some immediate remote changes. So I think it would be super interesting to directly compare with the same task the impact of online stimulation and offline stimulation. And I think naively, I always thought that if I want to have a good behavioral result, a good modulation, then I go for online stimulation. But even that, I think, is not true. Some of the offline studies that we see report some relatively robust results. If we try to interfere with the task online, then sometimes we don't get results. So I think this remains an open question and a puzzle when we might expect what. So, yeah.
I just had a very quick comment.
Yes.
How to control for variability in behavior in vision. In that sense, we're kind of luckier because what we try to do, we really try to work at threshold. So when we do that, and you really work on difficulty of the task, we really try to test everybody at threshold. That's when we really see the difference, including using 1 Hertz offline TMS.
Yeah.
So, but you know, we are particularly lucky that way. But so the idea here is really to try to test every single subject at their own threshold in whatever you're testing. So this is what we do. But again, I know it's more difficult in language, definitely.
It is difficult. I think one idea would be to at least consider the individual baseline level of task performance and take that into account. But I think if we're trying to really control for that, we might lose a lot of our subjects. And I mean, for very easy and automatic language tasks, most of our healthy subjects, healthy young subjects, they perform at ceiling level anyway. So we would need to target or to adapt the level of task difficulty to the individual needs. We've played around this with auditory discrimination a bit, so with speech and noise, and adjust the individual level of task difficulty to the individual hearing threshold. This is super complicated. We haven't combined that with neurostimulation yet, but I think this is a very good suggestion and could be a way to go, also to get stronger and more stable results, because some of our results may also be insignificant because the task is simply too easy. So we need to adjust the level and adjust it to the individual subject's abilities. Yeah, that would be a good idea.
Thank you.
Okay, thanks a lot. I think we should go for coffee. We have a lot of, obviously, possibility to interact later on. So there's now a coffee break and the poster break. Please go to the posters too. And we are coming back in 45 minutes, 1/2 past. Thank you.
And feel free to approach me during the breaks.